Olas Media. Olas Media presents Cannabis Enlightened. Now here's your host, Dr. Leroy Brady.
Hello and welcome everybody to another wonderful episode of Cannabis Enlightened. I am your host, Dr. Leroy. And this episode, as all our episodes, are brought to you by March and Ash. And today we have the distinct pleasure of having a very gifted scientist with us today. And he's going to talk about cannabis from a scientific standpoint, but I guarantee you from a scientific standpoint that you will understand what he's talking about and what the significance is between what he does and the cannabis that is brought to you through the cannabis dispensaries. And without any further ado, I'd like our guest to come on and introduce himself to us and tell us a little bit about himself. Reggie?
Hi, thanks for having me on. I'm Reggie Guadino, and I'm currently the Chief Science Officer of Front Range Biosciences, which was a fairly long journey through the cannabis industry and an educational path that I think you're alluding to. So my background is as a biologist is in molecular genetics and biochemistry, and basically what that really means is that I'm old enough that I entered at the very beginning of molecular biology. So when there were just things that were being discovered after DNA sequencing, so I've done all types of DNA sequencing from chemical cleavage all the way through current like nanopore or long read, high tech stuff. And we used to, I'm old enough that I used to purify my own restriction enzymes, right? So back when, before you could just, go to the local dispensary of molecular biology supplies and buy these things, we had to create them ourselves and isolate them from the organism. So I have a really deep understanding of DNA, nucleic acids, and so what I guess my bent was always trying to understand why, right? Why did genes turn on? Why was there expression? What caused that expression? How did that expression change? So, and early on, the only things you could really to look at, because there was an easily measurable outcome, was biochemical pathways, right? So things like sugar metabolism or antibiotic resistance, the genes, that turned on and off for those kind of things in bacteria and yeast. So my understanding is basically of how do these things interplay and control what ends up being what we see as a phenotype or an expression in a plant, right? So, or in any organism. So that's a long way of saying is, my training is in being able to understand the bits and pieces of genetics that end up making us as people or plants look and do what they do. So there's a big black box there, right? Where something happens in biology and we call that biochemical pathways, right? So in order to metabolize sugar, in order for us to, take in oxygen and bind that to the hemoglobin in our blood, those all happen because of biochemical interactions. So my training is in how do those things happen? What is responsible for controlling those things?
Okay. So when we talk about biology, let's give the listening audience who may not have been to college recently. When we talk about biology, what is that the study of?
So biology is typically the study of biological organisms, right? And typically biology is a catch-all term for, plants, animals, etc. So it's kind of a misnomer because I didn't study everything. I study specifically I've done the entire evolutionary chain. I've done bacteria, which is the single cell organisms, yeast, which is now a nucleated single cell organism, and then up the chain, right? So, but generally focused on plants and or bacteria and plants. So, but biology is basically the study of what keeps or what processes go on to make these things live and reproduce, right? So
Okay.
now that also is a huge, there's so many things involved in there, and my specialty, molecular genetics and biochemistry, is a very kind of directed look at the processes involved in genetics, in DNA replicating itself, being the blueprint that then gives us the RNA that gives us the protein. So I study that kind of aspect specific to biochemistry, right? And biochemistry again is a large term, but basically the things that allow us to respire, the things that allow us to convert food into glucose and starch. Those are all biochemical networks that are coordinated. And so I study my specialty is the coordinated regulation of the genes that give you these biochemical pathways that work.
How does that interact or intersect with cannabis?
So again, cannabis is a biological organism. So it has a bunch of processes that it needs to be able to do successfully, right? In its interaction with the environment, so that we can in fact cultivate it and harvest it for its products, right? So so how that translates is understanding the genome so that we can see what genes, and it's just very similar to human genome work, where after we sequenced the human genome, we were able to look for things like genes that were abnormal, that caused cancers, that caused disease, that were responsible for congenital defects, right? So so by understanding the genome, right, by understanding what genes make up that organism, and then understanding how differences in the gene sequence might either have a beneficial or a negative influence, and then being able to use that information for breeding purposes, for monitoring, and for even looking at the interaction between the plant and its environment, and also the plant in the soil. So so that's how a lot of the work that I do interacts specifically with cannabis, and the basis of the research and all of the basically the organization that I put together, because I have 12 scientists that work for me. So
Wow. And okay, so you have 12 scientists that work for you. Are they all working on cannabis, or they're working on other types of plants?
So the work that we do is specifically now directed towards cannabis. At one point, we did other plants as well, coffee, but we've shut down the non-cannabis divisions. Just coffee, taking over the coffee plant, trade in the United States didn't serve a lot of purpose, because there really wasn't a coffee plant trade in the United States. So it's because at one point there was a burgeoning local grown coffee industry that was coming up in Santa Barbara and some other places in California, and we were actually servicing some of that area, and it didn't go anywhere. So so we focused on hemp and cannabis. Well, I should say cannabis, which unfortunately is incorrectly broken into hemp and marijuana, right? So it's realistically the same plant, and so all of our work is focused on that for specifically improving cultivars, right? So ultimately, when you do any plant or agronomic research, it's for two things. It's to either understand or to improve, right? So and in order to improve, you have to understand first, right? So you have to lay the foundation. And because of the prohibition against cannabis for the last 100 years, right, there really has been no understanding of what the genome looked like of cannabis, and how the plant really interacted in a systematic way with its environment, which is pretty amazing because it's the same plant, very similar genetics. Obviously, evolution has caused some differences, right? But ultimately, the same plant that we see now, that's hemp and African varieties, or Tibetan varieties, vastly different in their geographies. Those all came from the same parent plant, right? So it was a progenitor cannabis plant that, through migration around the planet with humans, right, adapted in a way that very few other plants can, right? And so that's really one of the special things about cannabis, and that's what we're trying to understand. Why is cannabis so adaptable? Why can it take on so many forms and do so many things? And ultimately, that comes down to its genetics and the biochemistry, right? And the way that interplays with the environment.
Is cannabis brought to your laboratory as a leaf, or do you grow the cannabis within the confines of your laboratory? Exactly how do your scientists interact with the cannabis?
So it's a great question. So when I first got into the industry, right, we were the same people that are with me now were the basis of a regulatory testing laboratory structure, right? So we actually were a testing laboratory in the registered with the Bureau of Cannabis in California. And so a lot of our early work was regulatory based, and so samples were brought in, and through the whole regulatory network. More recently, as the chief science officer at Front Range, we're actually in agronomics, or an ag tech, and a breeding company, right? So ultimately, what I produce now is either seed or clonal propagates of certain varieties, right? And so we do it both on the hemp and regulated cannabis side. And so when we do a lot of our work, there are in fact field trials where either we have cannabis that is grown in one of our greenhouses, or in tents in one of our research facilities, or it's actually grown outdoors if it's hemp with partners, or in certain cases in Colorado, we actually had a regulated cannabis partner as well, trial on, I think it was 10 acres, a number of our varieties on 10 acres of outdoor land in cannabis. So, and then depending on where we are, so the cannabis that's grown and cultivated in California goes to our R&D space in California, unless it's hemp, in which case we can send it anywhere. The regulated cannabis that is grown in Colorado is tested at our facility in Colorado. So, so because of the boundaries and structures, we have to have more than one facility. But because also of the licensing, we are allowed to take it from an outdoor field and bring it into the laboratory, whether we grow it or not, as long as it's registered within the the metric framework of that state.
Okay, so a couple of things there that I wanted to ask you about. One is that you mentioned three different terms. You mentioned cannabis, hemp, and the word I use not too much, marijuana. When you say marijuana, are you also talking about cannabis?
So cannabis is in fact the correct terminology, right? So unfortunately, because of the regulatory structures that we have to operate in, right? So there is a category of a completely artificially determined category of cannabis called hemp. And that's regulated because it has less than 0.3% THC in the flower, right? Now, you heard me say regulated cannabis or marijuana, right? So, and those are the ones that you typically find in the adult use categories, or the the recreational categories, depending on the state, right? And those are regulated differently. And so because of that, even though it's the same plant, for me as a scientist, I have to treat them differently and handle them differently. And I even have to do research on them in separate establishments, right? So I can't do hemp research in my cannabis facility and vice versa.
Okay. So now I still want to go, that clears that up for me. How about, you talked about looking at cannabis and hemp from a cultivation standpoint. So are you looking at, or are you developing seed, or
Yes.
are your people looking at what grows from the seed?
Ultimately, right, in order for, let me go back even a little bit further. I hate to say it this way, but you know, agronomics, farming, right, is a commoditized industry, right? So at the end of the day, what has to happen is the most efficient production means possible, because the as you bulk up or as you produce more, the price that you can get goes down, right? So ultimately, then it becomes a margin-driven business, just like any other. I hate to say that, right? And ultimately, that is what will happen with cannabis, right? Like every other big farming operation is seed-based, right? So it's very expensive to go from clonally propagated. Imagine the labor it would take to plant 1,500 clones, right? But manually, because there's really no way to do it with a machine, right? As opposed to just driving up and down a lane with a seed thing that just punches every 6 feet, right? And it drops a seed, right? So suddenly now the cost of labor goes way down as you are now looking at scaling up production, right? So to do that, you have to have uniform seed, right? And uniform seed comes from a process where you do breeding experiments, you take some of that seed, you throw it out into the field, and you look to see what you've got. You pick the best ones, you breed the best ones, then you do it the next year again until you get to a population that where you have, if you throw 1,000 seed out, 995 will look identical, right? Because that's what you need to be able to guarantee the person that's planting that seed is going to be satisfied and be able to make his margins on his side, right? So there's a lot of agronomics that people don't ever see. When you go to the store and you get your strawberry, that strawberry is likely the result of 10 years of breeding and cultivation R&D before it hit the market. That's the truth, right? So cannabis, unfortunately, doesn't exist that way, right? So what we've done is we've hybridized cannabis a lot. And so because of it, when you do breeding, you end up with a population of seed that looks nothing like each other. And then from that point, you have to then take what you want and then breed down till you get to the point where you get that uniform seed population. And that typically, without heavy R&D and specialized kind of tricks, right, which have not yet been developed for cannabis like other plants because we haven't been studying it long enough, right? You're talking 8 to 10 years for cannabis as well, right, before you get to a stable seed population, right?
So are you saying it takes 10 years to perfect a seed that will grow fast and will produce the type of cannabis that is, I guess, profitable?
Yes and no, right? So right now, the cannabis industry exists generally as clonal propagates. There's very few people growing from seed. Generally, only breeders start with seed and they use that seed to pick some parents that will be the next generation donor, right? But what happens is when somebody finds something very unique and potentially profitable in the industry right now, they clone it. They make genetically identical copies because they clone the plant and then they root that cutting, and then they now get a plant that's identical to the one that was cut off of, right? Now, if you take a plant and you cut it 20 times and you get the 20 cuttings to stick, you now have 20 copies of that mom, and you can do the same thing. So you can see how you can clonally increase, right? You know, and it's just a matter of time before you can take one plant, you can make 100 moms, and then every 2 or 3 weeks, off of those 100 moms, if you're getting, 10 or 20 cuts, you're making 1,000 to 2,000 cuts a week, right? Or every couple of weeks, right? So this is how the industry currently operates, and you have And unfortunately, because it operates that way, it was the way a lot of disease got in. So I'm sure you guys have heard about HPLBD, which is like the HIV of the cannabis industry, right? So, and I say that way because it's an RNA virus. It is latent, and you don't know you got it until it's too late.
Reggie, is that some of the what can I call it? Disease? Is that a disease on the plant? Would that be found on plants that are grown like at home? Let's say if somebody wants to grow cannabis.
If you get a cutting from a dispensary, it could be.
Okay.
If you start with a seed, it is highly unlikely, right?
Okay.
So, and here's the nice thing from scientific research that, in fact, we've done some of it and others have done it as well. When you take diseased plants that have HPLBD and you breed them together, right? And you can look at the seed, less than 10% of the seed come out with the disease. So just breeding is a good way to clean from disease, at least for HPLBD. So,
your lab, can I call it a lab? It's a laboratory. Right?
There's a lab as part of the entire process, but yeah. So yes, we have a lab, but it's really a breeding and product development division.
Okay.
So, yeah.
Your breeding and product division. Yeah. Do you work more with clones or with seed?
We do both, right? So in order to produce millions of seed, right, you need a bunch of the same plant to be able, that's all bred to the same pollen donor, right? So yes. So our goal is to transition, hopefully, all of our clients to inbred seed that performs well for them in their circumstance, right? But for us to be able to produce that seed, we still have a need for some clonal propagation. I have a tissue culture lab where, we do the whole testing and cleaning and to make sure that our stock that goes into our breeding programs is clean, right? And just a few years ago, we were distributing clones because there were still so many people who in the industry were accustomed to clones, and even in the regulated cannabis, of California and Colorado, clone sales is still the way most people do it because nobody has scaled up yet. And so right now labor is manageable, but once you start planting 100 acres outdoor, there's no way you're planting clones because you're going to have a labor force, right? And of a sizable labor force, because you don't want two guys planting 100 acres, over a course of time, because then everything, you're not harvesting on the same time, because by the time they get done with acre 100, they're 2 weeks after and stuff is already growing, right? So
it sounds to me like if you are planting seeds and you're planting clones, your clones are going to come up before the seeds will.
So yes, but generally, right, you can plant seed sooner than you can put clones in, right? So clones, generally, you take them, you root them, and within a short period of them rooting, you're putting them out into the field, right? And so there's a potential there for clones that are not so hardy, or plants that are slow to start. They don't do quite as well as seed. Generally, plants that come from clones, in an outdoor environment at least, do not produce quite as much as plants that come from seed. And there are different theories about it, whether it has a taproot or not. So plants from seed will create a taproot. A taproot is the big central root that drives down and anchors, but also because it goes further down, produces a larger network of root than generally a clone, which starts as a little mass and doesn't have a taproot. And so most of the roots go out laterally, right? So at least that's the theory. And so generally you see plants that come from seed are hardier, but again, if you're doing things indoors, and that's the other thing, what is the method of cultivation? If you're cultivating indoors, some of these things don't matter, right? So, and now, and that's a whole other aspect of cannabis genetics, because we see a lot of things that are really sexy and sell very well, but those plants would never survive natural selection outdoors.
Stay with us. We'll be back with more Dr. Leroy after this quick break. You're listening to Cannabis Enlightened on the Olas Media Podcast Network.
American democracy's good, but we can make it better. The National Association of Nonpartisan Reformers includes organizations across the country who are working right now to build a better democracy by opening primaries, implementing safe, secure voting systems, reducing corruption, and increasing transparency. Listen to our weekly podcast, How to Win Friends and Save the Republic, to hear updates from the latest movements in the democracy reform space. Subscribe and learn more about us at nonpartisanreformers.org.
You're listening to Cannabis Enlightened on the Olas Media Podcast Network.
I noticed that most dispensaries have clones. They sell clones.
Mhm.
They do not sell seed. Is that because the clones are hardier or don't take as
No.
long to mature?
No, there's the time scales of cultivation from seed and clones is different, right? Because essentially, when you had to take a clone, the part to get to that size, that you've bypassed that. Somebody else has done that for you. So you're getting a plant that's X tall, right? But at that point, you still have to uppot it, and then you still have to get to a certain size. Then you still have to then flower it, right? And the time for a seed plant or a clone plant to go from the timing switch to finish flower is the same, right? It's just that to get to a plant that's 3 feet tall from seed takes you a couple extra months, but you don't see that because you got the 6-inch tall plant and then it gets to 3 feet tall in like 4 weeks, right? So realistically, when you look at the critical time period from timing switch to flower, they behave the same. It's just who grew the biomass before you got it? Was it you or was it somebody else? So the timing is so people look at it kind of the wrong way, because when you get something that's already done its first 4 weeks of growth, but you didn't do it, you think it's taking you less time, but it didn't really take less time, right? Because you still have, even when you take a cutting, it takes 7 to 10 days to root, and then it takes a little while to acclimate, and then it takes a little while for it to start growing, right? So somebody else has done that work for you, but the time still counts, right?
Well, the time still counts. However, if I'm understanding you correctly, if I get a clone, the clone is going to mature and provide me with the cannabis that I want, that I'm looking for, sooner than a seed would.
Okay, so if you started from a seed and then you took your own clones from that seed and you maintained your own mother stock, then you would have the same situation, right? So it's about the process, right? There's a process to make clones, and that's why clones cost between $7 and $10 a piece, right? So you're paying between $7 and $10 for a clone, right? And that's in bulk. Bulk seed costs $2 a piece, right? So already you're saving 3.5 fold right there. When you're dealing, it doesn't sound $3.5 doesn't sound, 300 fold, $5 extra doesn't sound like a lot, but multiply that by 10,000, right? So now you've just saved yourself $50,000 on. So it depends on the scale, right? Again, and so the home grower would probably never consider any of these things, right? And for them, going out and buying 6 clones a year is a good deal and gets them further. Yes, right. But again, it depends on how you look at it and what scale you're looking at it at.
Okay, so that if you are a cultivator, if you're a cultivator, you want to deal with seed rather than cloned.
If you're a large scale outdoor cultivator, absolutely. Right?
Okay.
So absolutely. And even larger scale indoor cultivators are now understanding that seed's also a way to go. A lot of people are starting to do this autoflower thing from seed, and autoflower can only be done from seed. Right?
Tell me about autoflower.
Autoflower is a plant that is photosensitive, photoperiod insensitive. So it runs a certain number of days, clicks on the flowering program without a light trigger, doesn't need light or whatever, right? So as long as you can grow it in 18 or 24-hour light all day long, but at day 50, if it's a 50-day program, it will start flowering on day 50, and then it will initiate its flower program, and X number of weeks later, you'll have bud. But because of that, you can't clone autoflowers, right? So now the only way to do large scale autoflower is to do from seed, and so that means that now you're constantly having to make seed because you can't clone them. And so if you're an autoflower producer, the whole thing that I said before about, for me, if I want to make millions of seed, right, I do it by taking the one parent that I want and making a whole bunch of her and taking pollen and then spraying all the pollen from the one male on all the clones of the one plant. Well, you can't do that. So for autoflower, you have to do continual seed increases. So if you take X number of plants that are male and female, you let them do their thing. Or you take some and you reverse them, you make them feminized pollen, and then you take that batch of seed and then you do the same thing with that batch of seed. So if you start with 10 seed, you might end up with 1,000 seed or a couple thousand seed afterwards. So to get to the millions, you have to take those couple of thousand now and then do the same thing.
The clients that come to you for clones or seed,
Mm-hmm.
which is the largest demand for, the clones or the seeds?
So it's a very interesting question. So on the hemp side, it went from clones to seed. A few years ago, we sold over 1 million clones, and then the year afterwards, it went down dramatically, and we sold a lot more seed. It hasn't switched on the cannabis side. On the cannabis side, we are seeing that the traditional method of cultivation, which is from clone, at least because most of cannabis is still indoor grown, and the outdoor cultivation that happens up in the Northern California, even a lot of that is still from clone, right? A lot of those guys are still doing from clone. So, and again, it's because nobody really has stabilized seed, right? And I think that gets back to what we were talking about before, where when you go to a dispensary and you see a variety called Blue Dream, right? And you can go to any dispensary and you can get that Blue Dream. But that's because somewhere the originator of that variety cloned it and handed it out to a number of people who were then able to clone it and then sell it, right? So, and until you can get to the point where you can take a seed batch and you can guarantee that that person will get the same kind of consistency, right, from every clone, right? So if you go buy 6 clones of Blue Dream, chances are you will get 6 identical plants in your yard, right? If you were to take 6 seed from a Blue Dream self, so that's where you take the pollen of Blue Dream and you fertilize Blue Dream with pollen that came from Blue Dream, right? You will get in your hand a batch of seed where you will have 6 or 8 or 10 different looking plants come out of that cross. And that's because it hasn't been genetically stabilized, right? And so that's right now the barrier to entry for seed in the cannabis industry, because people want that thing, and not everything will come out the same until you get to the point where I said 10 or so generations down, by doing that selfing process, every seed will start to look the same. In fact, there's a mathematical formulation that you can do, where you can figure out based on the size of the genome. And things like corn that have a very complicated genome, it takes them 18 generations to get inbred seed, right? Cannabis, because of the size of the genome, will probably be somewhere between 12 to 15 or 18 as well, probably, unless we can develop some of these new tricks that have been developed in other organisms, other plants, that break that down. Something called double haploid breeding, right? So where you basically take the gametes and you fuse them together and make a diploid, right? And then that diploid, because they came from two sperm cells, right? Or two pollen cells, right? Is 100% identical, right? And so in one generation, you've gone from a very heterogeneous genome to a very homogeneous genome. Homogeneous being mean they're the same on both parent strands are the same because of what we just talked about, and heterogeneous is what we normally have pollen and ovule and an egg come together, or sperm and an egg come together from a male and a female, right? So then you have a heterogeneous genome because you get one strand from the dad, one strand from the mom, right? So
so in closing, where do you see the future going?
Again, it's agriculture, right? So at some point, there will be vendors with large catalogs of very stabilized seed, and you'll be able to at some point go through a catalog and say, hey, oh, that plant has what we want, right? And so we were going to order a bunch of seeds of those things. Along the way to get there is kind of some of the work that we do is, you develop the breeding markers, you do the chemical studies that help you support the breeding marker work, right? So, when we're looking for minor cannabinoids, THCV, CBDV, CBC, some of these things other than THC or CBD, you have to find them chemically first. So there's a process of phenotypic hunting, right? Pheno hunting, where you have to go find the interesting things. You have to then study them, develop markers so that you can now take these traits and put them in any combination you want. That's where we are with a lot of our other agronomics right now, right? They've been studied for a long time. We can do those things with strawberries, blueberries, look at all the specialty varieties that come out of citrus. So, at some point we will catch up in the bioinformatics and the genomics and the chemistry, and we'll be able to breed cannabis for specific effect, for specific medicine, and for that have been augmented by those things that give you faster flowering time, higher yields, right? More disease and pest resistance, just like we've done with agriculture everywhere else. And so I think ultimately, we have a little ways to go because we are so far behind in the understanding, but ultimately that's where cannabis will be.
I'm going to make a point of asking you if we can check back in with you in a couple of months or so to find out, where you are, what have been the advances, successes, and I guess I have to say failures, because failures also lead you to successes in the future, right?
Yeah, absolutely. So I would love to come back and so agronomics, plant agriculture, there are more failures than success. You have to be comfortable with that. So
great. So can you, as we wind up and close, can you let our audience know again who you are and your particular company?
So I'm Reggie Guadino. I'm the Chief Science Officer at Front Range Biosciences. And we work on hemp and cannabis because it is a great plant.
And I think that, thank you very much, Reggie. I think that when many of the listeners listen to this, they're going to want to contact you or find out how can they get into the type of business that you're into, because it sounds absolutely fascinating. Absolutely fascinating.
Well, science is great. So if I had to say one thing, we do not have enough people of color who are in the science and or real hands-on development of cannabis, right? So we have all sorts of people doing everything else, but like when I go to conferences, you and I met at a conference, right? The distribution of people that we saw there was lacking, and so hopefully people like us will hear this and say, hey, I can do that. I'm a good scientist. I can do those things because the opportunity is there.
I think if you're there, they would be motivated. No question about it.
Yeah.
Thank you very much, Reggie. This
Thanks.
has been extremely interesting. This has been another episode of Cannabis Enlightened. I am your host, Dr. Leroy, and this has been brought to you by March and Ash. Thank you very much.
If you'd like to hear more Cannabis Enlightened with Dr. Leroy, be sure to visit us online at cannabisenlightened.com and subscribe wherever you download your podcasts.
Olas Media. Produced in San Diego, California.